Now let's talk about the genotypic methods used to identify a pathogen. So genotypic methods means that we're going to need some sort of DNA analysis, or if it's like an RNA virus that doesn't have DNA, we can also use some RNA methods to identify the pathogen. The first example of a genotypic method is something called FISH. FISH stands for fluorescent in situ hybridization. Whenever you see this word hybridization, that just means for like two things to hybridize or attach to each other. During hybridization, it's like two strands of either DNA or RNA are attaching or hybridizing to one another. So let me explain how FISH works. So let's say, for example, you had a potentially septic patient and you needed to figure out what kind of bacteria is causing this sepsis. So what you would have to do is take a blood sample from this patient. And of course, usually first we would do a gram stain. And then you might suspect from the gram stain, for example, if you saw gram-positive grape blade clusters, you would say, okay, it's probably Staph aureus. But you really need to confirm for sure before you start treating. So FISH is great because it's very rapid. It can be done in about 90 minutes. And so it's one of the most rapid tests. So how this is done is because we know the genome, the DNA sequence of Staph aureus, we can make these probes. And these probes are made out of a single strand of DNA. And this DNA will attach only to Staph aureus DNA. So for example, let's say the Staph aureus had a section of DNA with the sequence. I'm just making this up. It's not real. Remember, DNA is made up of A, T, G, and C bases. So we'll just pretend that this is the Staph aureus DNA sequence, part of the DNA sequence in Staph aureus. So what you would have is a probe that's made commercially. You can buy it. And the probe is the complementary sequence to this. So A would pair with T, T would pair with A, the G would pair with C, C would pair with G, and so on. So you would have this probe with the complementary sequence to the known Staph aureus DNA sequence. Now, attached to the probe, you have a fluorescent tag. So what you would do is you would take the DNA from your bacteria, you'd put it onto this blot paper, and then you would place this probe, the DNA probe, in with it, and they would hybridize. They would anneal and attach to one another. And because now they have attached, when you expose these samples to ultraviolet light, it will react with the fluorescent tag, and you'll see your bacteria fluoresce under the microscope. So FISH can specifically confirm if the bacteria is what you think it is. Now, if your DNA was not Staph, it was something else, these sequences wouldn't hybridize with each other, and the bacteria would never fluoresce, so you would have a negative result. So overall, positive result means you would see fluorescence. Negative result would mean there was no fluorescence. If you didn't see fluorescence, it's negative. This technique could also be used for RNA viruses. Now remember, RNA does not have T, instead it has U. So if you had a sequence like UACUUG, your probe would have a complementary sequence of AUGAAC. So these two fragments would still hybridize to one another, and you'd have the fluorescent tag here. So you could do this also for RNA sequences as well as DNA. So that's FISH. Again, it's a very rapid test, and it can be used to show DNA that hybridizes using fluorescence. Now, another technique that's genotypic is what's called pulse field gel electrophoresis. So how this works is, let's say that you have this bacteria or virus, and here's its DNA sequence. There are these enzymes, they're called endonucleases. Endonucleases, they will cut DNA at specific sites. So they're kind of like molecular scissors. So every single pathogen, it'll cut at a different site. So let's pretend like this is Staph aureus, again, just an example. And the endonuclease will cut the DNA here, it'll cut it here, and it'll cut it here. What you would end up with, you started with one big piece, but you would end up with 4 smaller pieces. Then what you could do is you would run your sample on something called a gel, using gel electrophoresis. Gel electrophoresis separates your DNA by size. Small DNA fragments go to the bottom, and bigger fragments will be at the top. And usually what we do is we put a known marker in. So gel electrophoresis can be used for DNA fragments and it can be used for RNA fragments. So let me just give you a random example here. Let's pretend like we suspected this patient had Staph aureus infection. So in our sample here, we could place known Staph aureus DNA. We could place maybe Staph epidermidis DNA. We could place Streptococcus pyogenes DNA. And then in our final well, we would put our patient. So we would cut the DNA from the patient with the endonucleases. So the Staph aureus we kind of know is a marker. We would put the marker here first. Maybe Staph epidermidis looks like this. Maybe Strep pyogenes looks like this. And then we put our patient sample. And we would see that the patient sample lined up, matched the Staph aureus sample. The bands were all the same size. From there, we can confirm, okay, yes, this patient has Staph aureus infection because the DNA fragments, after we cut them with this specific scissors, match up exactly with Staph aureus. So that's an example of why we would use pulse field gel electrophoresis. Now, by far, the most commonly used genotypic method, for sure, is polymerase chain reaction, or PCR. So PCR is used for DNA, and what it does is it amplifies or replicates, makes many copies of a very specific section of DNA. Similar to PCR, it amplifies DNA. We could also amplify RNA. If we're using RNA, we would use RT-PCR, or reverse transcription polymerase chain reaction. So for example, currently the coronavirus, when somebody gets tested for COVID-19, they use RT-PCR because these two, PCR and RT-PCR, are extraordinarily accurate. They're very sensitive, and it's relatively fast, and you could do a lot of samples at once with this. Other things that are diagnosed using RT-PCR or PCR is HIV, Lyme disease, HPV, tuberculosis, hepatitis. I have to say, way more than just this. Almost everything is diagnosed using PCR in this day and age. So let me kind of break down how PCR works. So let's say here again, you have a section of DNA. Okay, DNA is double-stranded, so you see these are the two strands that are attached to each other. So how PCR works is first you need the DNA. This is called the template DNA. It's the DNA that's taken from the bacteria or the virus. Then you need this enzyme called DNA polymerase. DNA polymerase can make copies of DNA. And then you need these things called primers. So primers are going to tell the DNA polymerase where to amplify. So how this works is, let's say that this is, for example, again, Staph aureus DNA. What we would do is we would order a primer, a forward primer, that only attaches to Staph aureus. It won't attach to any other bacteria. And then we would order a reverse primer that, again, only attaches to Staph aureus. So what we're going to do is we're going to make many copies of this one section of the DNA. So we replicate it and we make many, many, many copies of it. From there, we could do one of two things. We could either sequence this DNA. This might take a couple days, so it's not as quick, but we can determine the DNA sequence and see if it matches the known Staph aureus DNA sequence. Or again, we could use gel electrophoresis. So this gel, for example, is showing you here is the known, this would be what the known bands would look like for this specific pathogen. Okay, so these are the known ones. And then this would be the patient sample. And you can see the patient sample matches the known. So you can confirm, yes, it's positive the patient has this specific disease. Similarly, if you're using reverse transcription PCR, such as if you're diagnosing somebody with coronavirus, what they do is, coronavirus is an RNA virus, so RNA is single-stranded. Instead of using DNA polymerase, RT-PCR uses an enzyme called reverse transcriptase. And again, you'll need a primer. So how this works, again, is you have this primer, the reverse transcriptase will come in, and it'll make DNA copies from this RNA fragment. And from there, again, you can either sequence the genetic material, or you can use the gel electrophoresis. match up to the known coronavirus sequence and see if they align. Now, one of the other things you can do is after you've done either PCR or RT-PCR is what's called ribosomal RNA sequencing. Every single bacteria and eukaryotic microbe, not viruses, so all living organisms must have ribosomes because that's where proteins are made. So they all must have ribosomal RNA. And using the sequence of ribosomal RNA, you can pinpoint exactly what species of pathogen you have because ribosomal RNA is conserved. Every organism has it, but the sequence is slightly different for every organism. So it really can be used almost as like a microbial fingerprint to really specifically identify what the pathogen is. And this is by far one of the most common ways we can figure out a pathogen. It also can be used to identify a mixed culture. If you had multiple species of bacteria in one sample, you could find out kind of the percentage of each organism in that sample using ribosomal RNA sequencing.